Module 1, a review of cell structure and organelle function. In a previous section, I introduced the organization levels of the human body. So far, I have gone over the organism, an overview of the organ systems in the body, and what organs are. In Module 2, we will go into greater detail of tissues. So in this section, I want to do a review of cells, their structure, and how cellular organelles function. Remember, this anatomy course will focus on organ systems, organs, and tissues, so this section will be a quick overview of the smaller levels of organization. A typical animal cell looks like this. All organisms in kingdom Animalia have this basic cell type. Now, there really is no cell in the human body that looks exactly like this one. This cell has the basic features that you will find in the various body cells. Not all cells will have all of these structures, and some of them, like neurons, look quite differently. But all of these features are found in some cells of your body. For example, most of your cells do not have cilia, but the cells that line the airway to your lungs do. Cells are composed of organelles. The first cell component is the plasma membrane. It is the boundary of the cell, holding all the materials inside and controlling the entrance and exit of materials. The plasma membrane has receptors on its surface that behave like gatekeepers, controlling the entrance and exit of substances. The nucleus is surrounded by a nuclear membrane, which is called the nuclear envelope. It's made up of two pore-filled membranes which envelop the nucleus. Inside the nucleus is the majority of the cell's DNA. DNA stands for deoxyribonucleic acid. DNA is loosely packed in the nucleus unless it's time for the cell to reproduce, and then it tightly coils up. When it is spread out, it is referred to as chromatin. When it is more tightly coiled, we refer to the individual coiled strands as chromosomes. The nucleus contains the major information for the cell to function, so it is kind of like the control center. The DNA codes for all the proteins that the cell makes. Outside the nucleus, the cell is filled with a material called cytoplasm. It's a jelly-like material. The fluid portion is called cytosol, and it contains lots of dissolved chemicals that the cell needs for survival. Materials such as ions, proteins, and other molecules are dissolved in the cytosol to be used for processes such as breaking down sugars and fats, or the building up of proteins. Ribosomes are where the proteins are actually synthesized in the cell. Proteins play a major role in how a cell behaves in the body, so ribosomes with their protein production abilities are critical for cells. They may be quite small in comparison to the other components in a cell, but they're very important. Some ribosomes are attached to membranes, while others are not. We call the ones that are not attached free ribosomes. And that brings us to the next organelle I want to talk about, the endoplasmic reticulum. Endo means inside, and plasma refers to the cell's cytoplasm, and that's where it's located. Reticulum means network, so the endoplasmic reticulum is a network of material in the cell's cytoplasm. And here are where the attached ribosomes are located. There are actually two types of endoplasmic reticulum, rough and smooth. Smooth endoplasmic reticulum is comprised of a series of tubes used for transporting material from one part of the cell to another part. It's also involved in the production of lipids and carbohydrates. Rough endoplasmic reticulum also transfers material around the cell, but it is called rough because it has ribosomes attached to it. So the rough endoplasmic reticulum is involved in protein synthesis and also the intracellular transport of proteins. If a cell has a lot of rough endoplasmic reticulum, then you know it's a cell that's responsible for making lots of proteins. If a cell has a lot of smooth endoplasmic reticulum, you know it's responsible for making more lipids and carbohydrates. The Golgi apparatus is like the packaging plant of the cell. It receives materials, puts them in specific containers, and attaches a chemical address for these packages to go. Many chemicals travel through the Golgi apparatus before being secreted. If a cell has a lot of Golgi apparatuses, then you should expect it to be responsible for secreting material. Nerve cells or neurons have to secrete several chemicals, so they are filled with Golgi apparatuses. The secretory vessel that you see in the figure was produced by the Golgi apparatus. Again, it takes a specific chemical for secretion, puts it into a secretory vessel, which is like a small membrane-bound sac-like package. Then the vesicle pinches off the Golgi apparatus and moves through the cytoplasm to the inside of the plasma membrane, where it then releases its chemicals out of the cell. Some cells create several packages or secretory vesicles and hold them until the cell gets a specific signal. Then, when a chemical signal arrives, the cell is ready to release a larger supply of chemicals at once. Lysosomes are another type of vesicle. Lysis means a breaking down or rupturing or loosening of something. So lysosomes' main job is to break down lipids, proteins, polysaccharides, carbohydrates, and nucleic acids. Now that's a lot of different chemicals, so each lysosome has to have specific enzymes to do their specific jobs. But because the cell itself is made of lipids, proteins, polysaccharides, and these other materials, it would greatly damage the cell if the contents are released in the wrong place or all at once. In your text, it explains that this is the reason you can only survive without oxygen for up to about 8 minutes. That's because after that time, the lysosomes in the neurons of your body cannot stay together, and they release their enzymes into the cell, destroying all your neurons. Centrioles are located in the centrosome. The centrosome is a location of microtubule formation in the cell. Microtubules are made from proteins, spirally oriented into long tubules. This helps support the structure of the cell as well as its movement. Centrioles are a component in cellular reproduction. Cilia are also made up of microtubules. You may have studied microscopic organisms that have cilia, which behave as tiny hairs helping the cell to move. But you might be surprised to know that humans have some cells with cilia too. They're found in the back of your nose, down your trachea, which is the tube through which you breathe, and into the larger airways of your lungs. The cilia move mucus out of the airways and into your throat. Mucus is there to trap dust and other particles so they do not get down into your lungs. Microfilaments are for cell movement. They help some cells contract. When you flex your muscles, the microfilaments help the muscle cells contract and relax. And besides microtubules and microfilaments, you also have intermediate filaments in your cells for strength and support to help them hold their shape. Mitochondria are the powerhouses of the cell. This is where ATP synthesis occurs. ATP, or adenosine triphosphate, is the chemical in which cells store their energy. Now, this isn't the only place where ATP is produced. Some of it is made in the cytoplasm. However, most ATP formation is in the mitochondria. Cells with lots of mitochondria are cells that need lots of energy to do their jobs. When I introduced the nucleus, I mentioned that a majority of a cell's DNA is found there. But some DNA is stored in the mitochondria. We call this, of course, mitochondrial DNA, and it codes for certain proteins that are needed for a mitochondrion to do its work. And because these proteins need to be made in the mitochondrion, it also contains ribosomes. Remember, ribosomes are organelles for making proteins.